So now I'd like to invite the panelists for our next cancer immunotherapy panel to start coming up. And so this will be our panel on cancer immunotherapy looking to the future. So I think this past panel really did a great job of showing you where we are, the groundwork that's been laid, some of the key questions that we still have, which we have a lot of questions. And then this panel will really give you an idea of, one, some of the clinical work that is going on, and then where we're headed, and I think we have a lot of exciting topics to discuss. So this panel is moderated by Dr. Jill O'Donnell-Tormey from the Cancer Research Institute, and she'll introduce the panelists, and enjoy.
Thank you, Jessica. So first of all, I'd like to introduce myself. For those, I am not in the focused ultrasound field. I am the CEO and Director of Scientific Affairs at the Cancer Research Institute. And for those of you who don't know the Cancer Research Institute, next year we will be celebrating our 70th anniversary. So we are an organization that since its inception has focused on cancer immunology. And we've always had, back then, the hypothesis that your immune system could be used to treat, control, and potentially cure all cancers. It's been a long road, and I think there's something to be said about that to all of you in the focused ultrasound world. Things don't happen quickly, but I think if you fund excellent science and excellent people, and you stay the course, things can happen. And I think I'm very proud that CRI over the years has really funded some of the seminal discoveries that have gotten us to where we are today, which is about a decade ago, of I think now the world believing, as we always had, that your immune system could play a role in treating cancer. That said, you know, I think thanks, Tim, for a lot of the background in terms of the cancer immunity cycle and what the immune system does. And I think those in the field, I think, realize that about, I guess, maybe 10 or 12 years ago, the first checkpoint blockade antibody was approved for the treatment of melanoma, and after that was the anti-CTLA-4, and then since then, several, probably 6 or 7 anti-PD-1s or anti-PD-L1s for another checkpoint blockade. And this was an amazing validation of our hypothesis that, believe it or not, in certain cancers and in certain patients, if you take the brakes off the immune system, you are able to show that the immune system could actually lead to long-term and lasting responses in many cancer types. First of all, melanoma, and I think it was only when non-small cell lung cancer also showed the same thing that medical oncologists started to believe that, way maybe immunotherapy is not a flash in the pan. That said, I think there has been progress. There's been FDA approvals probably, you know, of these anti-PD-1 and these checkpoint blockades for probably over 20 to 30 cancers as we stand now. But not that much has really happened in the last decade, to be quite honest. And I think this is a telling point of that there was this great excitement, and I think everybody in the world decided we can combine everything with a checkpoint blockade and get responses. Well, that hasn't actually proven to be true. We know, though, we have a proof of principle now that in humans, when you modulate your immune system with checkpoints, and subsequently there's also another type of immunotherapy, CAR T cells, which have been approved for hematological malignancies, you can get some very significant responses, but really in a minority of patients. And in some cancer types, no responses at all. And it has shown that you can't just throw spaghetti on the wall and expect everything to stick. And so though a lot of different trials, there's probably at this point probably over 4,000 immunotherapy trials that are FDA approved and ongoing, and you can see there hasn't been a great deal of progress. In the time, there's only been one other checkpoint blockade, an anti-LAG, that has been approved, and I think we're at a point of saying, how do you rationally combine things? You have a baseline, but we need to mechanistically understand so that we can intelligently combine things that make sense. What will synergize with checkpoint inhibitors that are either happening in the lymph node or at the tumor site with other modalities be they chemotherapy, radiation, targeted therapy, focused ultrasound, but in a very, I think, mechanistically and hypothesis-driven way, where there is something to be said, not just because you have a drug, which I think a lot has happened in the biotech world. Someone has a drug and they think they combine it with PD-1 and they're going to have a success, and that hasn't proven to be the case. But with that said, I think focused ultrasound is a very likely candidate to be able to combine with immunotherapies, especially because it really is non-toxic. And that's always a problem with combinations. The more you start combining, toxicity and adverse events levels rise up and therefore limit efficacy. So I think we're at the point now that we're looking at this panel. Where are we going in the future? I think the last panel showed you it's a complicated system. Not only is the immune system so complicated and involves a ton of different types of cells, not just T cells, but you have B cells there, and you have macrophages, and you have dendritic cells, and we're finding, as I think was pointed out in the first panel, it's not just the immune systems that matter. What is happening in that tumor microenvironment? The non-immune cells, we're finding that other things affect it, metabolomics affect things, the microbiome affect things, so these are all multi-component systems. And then you bring in focused ultrasound. And I have been collaborating with the Focused Ultrasound Foundation and Jessica for, I guess, it's about a decade, it's about 10 years now, I guess. It's been a long time. It seems like a long time. And I still can't get it straight, all the different types of focused ultrasounds that there are, and how they're used, and what they do. And I always, back 10 years ago, I said, this sounds like it's a great thing to combine with immunotherapy, but we don't even know what focused ultrasound does to the immune system. So how can you intelligently say, let's do this? So I think great progress has been made thanks to the foundation here in funding some very, very basic knowledge, but you can see it's still complicated. I think in this last panel, we don't really know exactly what's happening of the effect of focused ultrasound, various different types, on your immune system. But that said, I think we're at a place now that there are clinical trials using focused ultrasound, and we're going to hear that in this panel. And I think I'm going to allow each one of our panelists to tell you a little about what they're doing, but I'm going to throw out a provocative question here after we hear what they're doing. What is the role in doing kind of preclinical translational researches versus reverse translation? Are we at a point now that we have clinical trials using focused ultrasound? Should we be, switching our focus to doing that analysis on these very precious patient samples and finding out responders and non-responders when we're using focused ultrasound, and then analyzing that so that we can get hypothesis that we can bring back to the lab and test, instead of doing the opposite, which we hear the problems with different mouse lines, with different, how are these preclinical models really telling us about what's happening in the human system? So that's a provocative question. I'm going to have I'm giving you a heads up, all you panelists, in terms of answering that question, but I think it's an important one. But with that said, I'm first going to now introduce our panel. We have, virtually, we have Patrick Dillon, and he is from the University of Virginia. We have in person Natasha Sheybani from the University of Virginia also. Alayna is here from Virginia Tech. John Groot is virtual from UCSF. And then we have Peter, he goes by Peter, right? Peter Wang from the University of California, San Diego. So I'm going to start by having each of you maybe give a very brief summary of the work you're doing, and then we'll get into some questions after that. So I think we'll start. Patrick, would you kick us off?
Well, Jill, thank you so much. An honor to get to go first. And so I'm a breast medical oncologist. So as you were just alluding to, the clinical side of things is exciting. So at the University of Virginia, and I'll try to keep it as brief as I can, we have announced at this meeting that we have been doing what you just said, the idea that you can combine a checkpoint inhibitor. We combined a PD-L1 inhibitor, pembrolizumab, with focused ultrasound, specifically in metastatic breast cancer patients. And we had been looking at the clinical outcomes, and we're just wrapping up that trial so that we can do exactly what you're suggesting. Look back now at those responders and non-responders, try to see what are the clinical differences, because for better or for worse, we took sort of all comers, triple negatives, HER2 positives, ER positives, And as we've been learning, there are huge differences amongst the different subtypes, the elasticity in these different tumors, the inherent underlying amount of immune recognition of the tumor in the first place is different. But we're now trying to go back and look and see, well, are there differences in some sort of biomarkers in the baseline tumor-infiltrating lymphocytes, trying to look now at the cytokine signatures and some of the transcriptomics. And so the excitement is that there are some patients who, when given, when a metastatic breast cancer is treated with focused ultrasound and pembrolizumab, there seems to be some evidence of response, but there's also some non-responders. And so just like we've heard at this meeting, we would love to try and increase the response rate and figure out what are the best opportunities for optimizing the device, the energy delivery. We talked about whether full thermal ablation or subthermal, or maybe even low dose ultrasound, or maybe using some sort of sonosensitization and a lower dose focused ultrasound. All those are great questions, and we will have some results. Also, at this meeting, Dr. David Brennan, one of my colleagues, went over our other ongoing trial, which is focused ultrasound for early stage breast cancer, and we're trying to look at combining that with gemcitabine in a low dose as a modulator of the myeloid-derived suppressor cells. So trying to deplete out some of the myeloid cells, which are a major problem in our breast cancer samples, to see if that can help further augment our response to focused ultrasound in earlier stage breast cancer being treated prior to a lumpectomy or mastectomy. And of course, we are doing a lot of the same transcriptomics and cytokine signaling and very careful spatial cellular microenvironment analysis. So that's the excitement, and maybe I'll cut it short so that everyone else can get a chance to tell us what they're up to.
Thank you. Thanks very much, Patrick. So, Alayna, you work with animal models, yes? But not mice.
Our lab primarily, I work with Dr. Joanne Tui at the Animal Cancer Care and Research Center at Virginia Tech. So we are using the canine model of osteosarcoma, so spontaneously occurring osteosarcoma in canines, which actually serves as a really valuable comparative oncology model for humans. And we are treating osteosarcoma with histotripsy, and we are looking at the immune response associated with the histotripsy ablation of the tumors. And I guess, as you just mentioned about kind of the reverse translation approach, we are now also starting to do orthotopic osteosarcoma mouse model studies and in vitro studies to get a better understanding of what's going on with the immune response.
Okay, thanks. Okay, John, I know you're in brain.
Yeah. I'm John Groot. I'm a neuro-oncologist at UCSF. So I am involved in multiple different approaches using ultrasound to treat primary and metastatic brain tumors. So there's, just as a really brief background, GBM in particular hasn't had any drug approvals in several decades, and we've been recently trying checkpoint inhibitors. And to your point earlier, not only do they not work, but they actually may even worsen outcomes slightly, based on several randomized phase 3 clinical trials. So we have a lot of work that we need to do, and I think I would agree that ultrasound may be a potential combination partner that could sort of overturn and make these ineffective therapies more effective. So it has multiple uses for our field. One of them, that it's not therapeutic, but more diagnostic. It's difficult to get access to brain tumors without having to go through a major surgery. So there's lots of work being done to look at ultrasound to disrupt the barrier, release ctDNA, release other biomarkers that can tell us more about the biology of the tumor without having to take patients to surgery. We're also doing clinical studies in combination with checkpoint inhibitors using a neoadjuvant approach. And that also gives us fresh on-treatment tissue to better understand what's happening with the tumor, both from the ultrasound itself as well as the checkpoint inhibitor. And then lastly, we're also doing, as Patrick was mentioning too, doing brain metastases work. And so that's another area of critical unmet need, and trying to enhance both cytotoxic as well as immunotherapy delivery to patients with metastatic brain cancer. Looking forward to the discussion and talking more about reverse translation and the challenges facing the field.
Great. Now, Peter, you're working in a very different space, inducible CAR T cells. So can you tell us about that?
Thank you. So yeah, I'm a professor at UC San Diego in bioengineering, and we are mostly working on genetic engineering for like CAR T cells. So these CAR T will have a gene which will be controllable by ultrasound. So we can only turn on the CAR expression when the ultrasound is shining onto the cells. So therefore, we can control the activation of CAR T only at the tumor site. Because right now there is an issue that, CAR T is very successful for blood cancer, but if for solid tumor, there is so-called on-target of tumor toxicity. Therefore, it's very dangerous if we directly use a CAR T for solid tumor. They will attack the tumor. At the same time, they will attack some other organs, expressing even minor antigens. So with all CAR T, we can introduce into the body, but they will not be having toxicity against normal tissue. Only when ultrasound is activating them at the tumor site, they will become effective. So that's one end of the work in the lab. We are recently also trying to use a vaccine approach to modulate the tumor cells so that they can produce like clinically validated antigen to serve as a target. They can recruit the synthesize the CAR T, and therefore we can also attack the tumor in the local region. So these are mainly the two ends of work in the lab. I hope we can share more during the discussion.
And last but not least, Natasha.
So hello, everyone. My name is Natasha Sheybani, and I'm an assistant professor of biomedical engineering at the University of Virginia. And I think I've had the good fortune of kind of growing with this field. I entered the field when it was far more nascent than where we are today as a graduate student, and just recently launched a lab that is really centered on ushering focused ultrasound into the era of precision immuno-oncology. And so the way that we're approaching that is by thinking of the major strengths of focused ultrasound as a non-invasive, non-ionizing technology. And I believe that that is best served by coupling this technology with non-invasive means for surveilling the approaches by which we can actually better deploy our immunotherapies with FUS. And so I mentioned having grown with the field because for several years now I've kind of gotten a front row seat in the process of understanding what our burning questions are. And that helped me develop a very quick appreciation, as Dr. O'Donnell Tormey alluded to, for the dynamic and vast parameter space that's in part underscored by the sheer versatility of the technology. So we have been doing immunomodulation work, drug delivery work for years now, but we're starting to take a step back to understand how we can leverage tools like liquid biopsy, molecular imaging, in order to better contextualize the nature by which focused ultrasound can help potentiate immunotherapies. So we're doing a lot of work with checkpoint inhibitors right now, starting to move into the engineered T-cell space. But in doing so, we also have the good fortune of working with clinicians like Dr. Dillon and others at UVA who are really pioneering in the space of clinical translation of the technology. And so we did some work early on exploring a paradigm combining gemcitabine and focused ultrasound thermal ablation that's now in the clinic. And so just to sort of jump the gun and answer the question you posed to us, I will say that I am a firm believer in reverse translation and its ability to really better inform what we should be doing at the bench side. And I think we're probably going to talk about why that's very necessary in the way of understanding how our preclinical models recapitulate what's going on with human disease, and also because so much of this parameter space probably has to be parsed out empirically, I think exploring things like sequencing of therapies, dosing, and otherwise will be best informed by going to humans where we can, and then ensuring that our preclinical paradigms are, in turn, helping to inform the mechanistic underpinnings of those discoveries. So I'll leave it there for now.
So where do we think, obviously there are certain tumor types that I think seem most amenable probably for use of focused ultrasound, and I think many of you are working in those. So I'd love you all to kind of opine on what is the human model that we think we can get the most bang for our buck, and that there's rationale to go into the human in the first place, and be able to also access the tissues that we need to do the analysis that will help us push the field forward. Anyone like to jump in first? Patrick, you look like you're dying to answer, so go ahead.
Sure. Well, I think we should be cognizant of the fact that we've been able to use focused ultrasound in most areas of the human body. Now, aside from the lungs, we have found that we've been able to access even deep tissue. We've had some success in pancreatic cancers, which are notoriously hard to access just because they're deep. We've had success now in some of the hepatocellular carcinomas. There's a nice presentation at this meeting. And so the other inherent question is, since we can access renal cell and prostate and pancreas and skin, and my interest in breast and brain, it's almost more of a question of what are the unmet needs and what are the unmet needs from an immunologic standpoint. And so melanoma, we were, as you were saying, 10 years ago, we were excited. This the flash in the pan of the immunotherapies, and we were seeing some of these melanoma patients achieve long-term disease control. But still, after 10 years, we really have not crested the 50% mark on controlling melanoma with immunotherapy. And so I think trying to better optimize our control of melanoma. So there's an unmet need for, yes, some melanomas are responsive to immunotherapy, but we are still missing a huge chunk of them. And so that's a particularly appealing clinical space to work in the melanoma space, because a lot of those cancers are immunogenic, but yet we haven't entirely optimized that. And so I think it's a great opportunity, since many melanomas do have easily accessible nodal lesions and skin lesions that focused ultrasound we've been pretty good at targeting in the clinic. Obviously, my own bias is breast cancer, because there's huge unmet needs in the breast cancer space. Immunotherapies have really only worked in a subset of triple-negative breast cancers and have not achieved at least a clinical approval or clinical success in the 80% of breast cancers that are ER positive. So there's a huge unmet need. There's a tumor that's close enough to the skin surface and easily targetable by existing devices on the market, so we don't have to necessarily generate entirely new devices to be able to go after breast cancer. So I think that's a great space to continue to work in. I think there's growing interest in some of these, the brain space, and obviously we've had several different meetings just specifically dedicated to both brain tumors and glioma, and the ability to try and target that, and we can let John talk about that a little bit more too. So I don't think that we necessarily have to limit the future development of focused ultrasound in the clinic. I think there are applications as long as we couple that with very close analysis and understanding of what's happening immunologically, and appreciate that there are wide differences immunologically in the different tumors. We've talked about microbiome differences. We've talked about differences in the power. So I think we just have to be thoughtful and perhaps not group too many heterogeneous patient types together in one study. So I think that's directions forward.
So anyone else? I guess I'm wondering, is it clearly melanoma is the poster child for immunotherapy in terms of having the most successes. So do you go after that? There's a lot known about that. There's an awful lot of data there. There's a lot of information versus in one of the cancer types where really immunotherapy has failed. I mean, you could take prostate, you could take pancreas, you could take brain, ovaries. What is there an advantage going to one where you're looking for the incremental? If you can, if checkpoints on themselves are going to go 50% with melanoma, say you have combined checkpoints, you can get a 50% response. Is it best to start there, or is it best to start in a cancer type where there's zero response? I would be interested in people's views on that.
I think it's great to know the baseline that you're starting at. So we understand a little bit about what the checkpoint inhibitors do in melanoma. Obviously, we don't entirely understand the resistance to checkpoint inhibitors, but I think it's wise to start with some baseline understanding. So I think it's great that we've done a lot of work in the melanoma space already.
Okay. Others?
Yeah, I would agree. I think Obviously, I'm completely biased with diseases in the brain and cancer specifically, but I think that as long as we design studies where we're able to learn from what we are doing to the tumor, then I think we're going to be able to advance the field. I think there can be a tendency sometimes to just kind of throw a drug at a tumor and see if it responds, and there's certainly drug development pathways where that makes sense. In brain, at least, where there is no response, I think designing studies where you're able to give ultrasound or checkpoint inhibitors or other therapies, or even cell therapies, and actually analyzing the tumor post-treatment to understand what the impact of your therapy was. I just think those are really critical studies to have at this very, very early stage where we're still trying to figure out what's the best approach.
Is it easy to get serial biopsies in your patients onto study?
No, but we can do it once. So we can do it on treatment study. But again, that's where I think the opportunity is to do ultrasound prior to treatment, so that you can at least analyze what's happening, from the ctDNA and or cells released from the disruption.
How about CAR T cells? What do you think there?
Yeah, I want to echo a little bit on that aspect. I thought it would be nice to have a systematic way to essentially collect the tumor tissue after ultrasound treatment, on the different modality, HARF or histotripsy, and then we can track on time, what kind of transcriptome, profile has been changing by sequencing. And therefore, we know what kind of antigen will be produced upon treatment, and therefore we can design more specific synthetic CAR T cells targeting, specifically on the different stage of the tumors. I guess probably these will apply to different tumor types as well. Obviously it will have difference.
Is it easy to design a study where you can give focused ultrasound first at baseline, take a tumor sample prior to then giving checkpoints, and then tracking, like knowing what focused ultrasound has done at baseline? Then you come in with a checkpoint, you follow up with a biopsy mid-treatment or something, or maybe it has to be at the end of the treatment, and see what changes. Does the FDA allow those type of studies to be done from the clinicians here?
So maybe I'll jump in and answer that. It depends really upon where the tumor is as to how frequently you can go back and rebiopsy from an FDA standpoint and from a patient tolerance standpoint. Mm-hmm. So having designed several trials over the years, we all want lots of biopsies, and serial biopsies do help us track with time. And it's beautiful sometimes because you can see incremental change over time as you go through a study. So in something that's cutaneous, like some of the melanomas, it's fairly easy to go back and sequentially rebiopsy. In breast cancers, we've done that as well with some success, and there's a little bit more grumbling, and patients are a little bit less happy with us when we ask them to biopsy multiple times on our breast cancer studies. But then you take a study in pancreas or liver or sometimes the prostate studies, and you add several more layers of risk to each sequential biopsy that you do. So, where it's easy to control any minor bleeding from a biopsy, it's easier to do those sequential biopsy studies. But even in breast, we get big, huge seromas from time to time with even the smallest little biopsies. So it's not entirely trivial, and you have to really have an excellent justification to the FDA and to your IRB for why you want to get those serial biopsies.
So are we, I think I know the answer to this question, but are any of this focused ultrasound studies, are they all focusing on combining with the known and approved immunotherapies right now, with the checkpoints and with CAR Ts? But are we ready to say maybe we move beyond that? There are a lot of other immunotherapy agents that are still investigational. Is there rationale for that? Natasha?
I would say certainly yes, but if you look at the composition of the literature and where a lot of people have focused effort to date, it has definitely been on the approved, established checkpoint inhibitors, namely anti-PD-1, anti-CTLA-4. We've made some forays into like the innate immune checkpoint blockade space, but there are now approved drugs in that space as well. All of that being said, I think a lot of us are starting to think about, the novel immunotherapies, exciting sonogenetically engineered T cells being among them, vaccination approaches were alluded to, and your question about rationale, I think, is a fascinating one, because at the end of the day, that's sort of what brings us back to this notion of what do we leverage in order to decide on rational combinations. And as Dr. de Groot mentioned, as much as we can sort of take drugs and throw them at the tumor and combine with focused ultrasound, this is where I think we're really going to have to deploy systematic efforts to know where something like a novel therapy makes the most sense. But I will also add to that, if I may, that where we have placed a lot of concerted effort into deploying the more established drugs, like classic checkpoint inhibitors, there's really an opportunity to learn, because of the wealth of data that we've amassed as a field, how focused ultrasound, where and when it should be deployed, but also how it has aided those efforts to date. And so I don't think that, I should say there's really an opportunity to do a deeper dive there and sort of fortify a role for focused ultrasound as well before we move forward with other approaches.
Now, Alayna, are you focusing in, and I mean, I love your model because it's not an inbred model, right? These are much more closer to regular patients getting cancer. I mean, and so have you focused on, and with animals, maybe you can get more tumor biopsies, can you? Or is that still not likely? They don't?
No, so for our canine osteosarcoma patients, we actually, even though they're dogs, we can't take like serial biopsies just because of the risk of pathological fracture. And currently we're at the point where we're partially ablating the tumors and then resecting 1, 3, or 5 days post-treatment. So these patients are still undergoing standard of care limb amputation surgery because they are really painful, and owners don't want their dogs continuing to be in pain. So I think that's probably a future approach, to look at the role of focused ultrasound in alleviating the bone pain associated with osteosarcoma. But we have talked about the logistics of trying to do serial biopsies.
Bone biopsies are
those in themselves are difficult. But it's just too risky, and the ethics of it aren't rational.
How about the view, I mean, obviously, so much of the field is focused, and immunotherapy is focused on T cells. How much have we looked with focused ultrasound on the other components of the, obviously the innate cells, but also how about macrophages and such? Do anyone have data on that they'd like to share in terms of where that would be? Are we looking at that? Or are we still just focused? We're so T cell centric. I feel the whole field is so T cell centric, but I think we have to go beyond the T cells. Anyone?
That's a great question. I totally agree. The integration ultrasound and synthetic biology will open a lot of opportunities. Particularly, you mentioned about not only CAR T cells but also macrophage, NK cell. Those can all be engineered with specific gene circuit sensitive to ultrasound, so you can precisely control.
You can engineer them for your focused ultrasound activation. All those cell types, not just T cells.
Because those are genes you can introduce into different cell types, right? So those are all doable. Actually, you can also do that to the tumor cells so that they can secrete or produce the clinically validated antigen or cytokine, and therefore you can design specific synthetic cells to target them.
How close are you to the clinic with this?
That's a good question. I think for CAR T cells, we are pushing toward the clinical trials, actually discussing with FDA. But for the other types, it's still probably at the early stage,
No, okay. I had a question and it just went out of my head. What do you feel, I mean, this panel is supposed to be about the future. So where do we think, I mean, I guess I'm going to throw out something. What about window of opportunity trials, where you can get tons of tissue? I think this, Pam Sharma at MD Anderson certainly has been a champion of this in the prostate space, where you can come in with checkpoints early on, beforehand, before you, people that are early stage going in for a prostatectomy, but you still give checkpoints ahead of time, and then you go through the prostatectomy and you can really see what the checkpoints are doing at the tumor. Is that something we should be doing with focused ultrasound in humans to really get a better sense? Obviously the tumor microenvironment is where the action is. I think we need to understand what's happening. We heard in the previous panel the spatial alignment of all the different cells in a tumor microenvironment. And then I think also with all the multi-omic analyses that we now have available, to be able to match that with blood to see if we don't need the serial biopsies, can we get signatures that we can find in the blood that are going to tell us about responders and non-responders. So anyone's ideas on window of opportunity trials, and is there another tumor type besides prostate cancer where you could do that?
Yeah. I can start real quick. That's actually the study that we designed, and will be starting very, very soon. It's a neoadjuvant checkpoint inhibitor given to patients with recurrent glioblastoma prior to surgery. So we will have all of that on treatment, tumor tissue for analysis, blood, imaging, all of the different omic analyses downstream, immune monitoring and such. So that will be starting soon. And I believe those are the kinds of studies that we need to do. Again, we can make arguments to do the same kinds of studies with T cells or engineered T cells or other kinds of therapies. But yes, we're heading in that direction.
And are you giving, in that study, are you giving the focused ultrasound with the checkpoint simultaneously? Yes?
Correct, yes.
Yeah, because you have to start somewhere, but I know, scheduling and dosing is going to be a real important thing going down the line, but you have to start somewhere and get some baseline to begin with.
Exactly.
Patrick?
Yeah, ditto, really. The current study that we are doing in breast cancer in the early stage, patients would be getting curative lumpectomy or mastectomy. Based upon work that Natasha and Tim Bullock from the last panel just described, we are going to give a single dose of low amount of gemcitabine and sneak in one treatment with focused ultrasound right in between the baseline breast biopsy and the lumpectomy or the mastectomy. And in order to do that in a timely fashion, we are doing most of our focused ultrasounds when the patient is in getting their sentinel node procedure, which is usually done before the definitive lumpectomy or mastectomy. So timing is really challenging, and it's sometimes hard for a patient to wrap their head around, oh, I've got to hurry up and get my definitive surgery, but I've also got to sneak in time for this other treatment. And so that's been our biggest clinical obstacle to doing these window of opportunity trials. It's really just been trying to find the timing that works with the patients and having time with our mammographers or our surgeons to fit everything in before the window sort of closes. Because with cancer, obviously, we don't want to wait too long before we proceed with a definitive surgery when we're in a curative setting. So I think it's huge and it's tremendous, especially when there's cancers where there's time. So prostate is another great example where most prostate cancers are slower growing. That window of time should be a little bit wider, just like a lot of the slower growing breast cancers. Our time window is wide enough that the FDA looks favorably upon window of opportunity trials in those kind of settings.
Anyone else? Zoe, what would, at this point, think of what you view in the future? What are we have a question. Sorry.
Okay,
Go ahead.
No, I don't have a question. I'm just going to make a comment about your last point about what are the opportunities for future. And I like to bring into the conversation the opportunities that are created by ultrasound with respect to targeting specific oncogenes and gene therapy. So I think there could be some unique opportunities there using gene delivery, using ultrasound, in order to target specific oncogenes that is already a growing understanding on how they can modulate the tumor microenvironment and make it anti-inflammatory. So there is a golden opportunity there to use the strategies that we have developed in this community for gene delivery to impact the tumor microenvironment in that way that eventually might make it more amenable to immune checkpoint inhibitor and therapeutic strategies like CAR T cells or other.
Well, thank you for that.
immunoadjuvants.
I guess we didn't really talk about how focused ultrasound is a delivery mechanism. I know it's been a drug delivery, but you're saying for gene delivery. And anyone here have experience with that? Natasha, have you worked with them?
Yeah, we're certainly working in the drug delivery space right now with therapeutic antibodies mainly. But that comment, which is very much appreciated, it brings the conversation full circle in a way, because we were talking earlier about how to decide where to leverage FUS, and it's a multi-pronged approach, right? We now know that we can modulate the immune system, and we're trying to figure out how exactly to wrangle that. But at the same time, we've thought about FUS classically as a modality for potentiating drug and gene delivery, and that tends to be still a major bottleneck for a lot of our immunotherapies, independent of the baseline for the immune landscape. So I think it's a very salient comment, because fundamentally identifying those opportunities where the chief bottleneck is a delivery problem, I think presents a really ripe opportunity for using FUS in a very deliberate manner. In thinking about that, I think there have certainly been challenges with figuring out what approach to take, right? I think we have a slew of different methods that we have access to, and the prior panel did a really nice job of summarizing those, but often the methods we're employing will allow us, say, a spatiotemporal snapshot, to kind of pick a time point or a location, or sometimes lose that spatial information. And so thinking about ways that we can incorporate functional imaging and complementarity to some of our drug delivery approaches is also going to be powerful going forward. And we're well positioned to do that as a field that has already adopted imaging as, an adjunct to much of what we do. So.
We have another question.
Yeah, I'd like to bring up another point in this discussion, in this reverse translation that we all really favorably look upon. There is the cost, especially the cost of the checkpoint inhibitors. If you look at those combinations together with FUS, they are usually at a level you are looking at around of the order of $70,000 per patient, and these are at levels that are very hard to include in regular grants, so there is a real difficulty in connecting with pharma and to getting support. And this is one of the issues that I would like the panel to think about and see how we can, together with the Cancer Research Institute, really address this together with pharma.
Yeah, so I know at CRI, independent of focused ultrasound, we had started what we call our clinical strategy, which was called the Clinical Accelerator, which was really to be an academic industry incubator where we would use academic ideas and hypotheses of what drug combinations we should get. And then CRI has actively gone out to negotiate with pharma or biotech to gain access to those agents and also allowing us to combine. We've been quite successful. We've had great partnerships with Bristol Myers Squibb, with AstraZeneca, and in many times, obviously in these things they do have kind of a right of first refusal. But I think they also realize that they're getting another shot on goal. They're getting great without paying consulting fees. They're getting this brain trust coming up with the best ideas of potentially how to use their agents. There's no guarantee. It's a problem, and I think, and pharma does obviously have a lot of investigator-initiated trial opportunities where you can submit your actual ideas directly to them, and in many times they provide no funding, but they'll provide drug. But it's an ongoing. It's not easy, and I understand that. And again, in a combination therapy, it might be a better idea to go where it's already been approved as a standard of care, where you won't have to pay for the drug. It will be paid for, and then you're combining your focused ultrasound like on top of that in a trial. But I understand the difficulty of getting drugs, and it requires a lot of work and relationships to gain access. But thank you for that.
Okay.
And don't forget that CTEP has 2 checkpoint inhibitors available that you can access through that pathway as well.
Yeah. So CTEP too. Yes, through the government too is another source of.
Can I add a quick note?
Sure.
Just to follow up the gene therapy and ultrasound integration.
Yes.
There is actually a conference or symposium organized by Mikhail Shapiro and several colleagues and myself. We call it International Symposium on Biomolecular and Ultrasound Sonogenetics. Essentially try to integrate the synthetic biology, gene therapy together with ultrasound, which will occur in December at Caltech. If you have time, please check it out.
Oh, that's great. That's great. So certainly the field's moving in another direction with synthetic engineering. It's wonderful. So synthetic biology and focused ultrasound come together. It's something that wasn't around 10 years ago, for sure. So we're coming up at the end. I don't think I have any questions online. So I guess we'll give everyone a chance of what is the most exciting and probably realistic opportunities of where we think focused ultrasound will have clinical impact in the next, I'd say, decade. So we'll start at it. Alayna?
So I think with our canine studies, we've demonstrated the feasibility of treating the primary bone tumors in these patients. Our preliminary data indicates that we are modulating the local tumor microenvironment, and exactly what is happening requires much further investigation. However, I think it is promising, and our future studies will look into this, and probably our next steps are actually just treating and leaving the tumor intact and advancing our devices to actually be able to fully try to ablate tumors and looking at the biomechanical integrity of the leg after the ablation of the tumor, to ultimately have this as a non-invasive approach to remove the primary tumor and hopefully stimulate an immune response to target metastatic disease.
Thanks. Peter?
Mentioning about synthetic biology, genetic therapy integrated with ultrasound, I think another one which could be also becoming a trend in the future would be the next-gen sequencing, genome, epigenome sequencing upon ultrasound stimulation, so we can really understand inside and out what's going on, to the tumor cells and immune cells, and this integration probably will be really helpful for us.
Natasha?
So maybe I'll just sort of spin off of the last two panelists' comments, because I completely agree. We're in an era now where we have such advanced tools, and with that comes so many exciting opportunities to mine deeper than we ever have before, whether that's at the level of molecular methodology or molecular biology methodology, I should say, or in the realm of imaging. And so I think it's very appropriate that one of our next panels is going to be machine learning and FUS, because as much as we didn't talk about this, we're rapidly moving as a field, despite our nascency, towards an era of big data, and with that will come the challenge of reconciling what's going on and enabling pattern recognition within those data. So I look forward to a future where we're going to be able to actually leverage AI-driven approaches through our systematic interrogations that we're doing now in order to actually do predictive modeling and enable precision medicine.
John?
Yeah, I think just very selfishly thinking about glioblastoma in the future, the tumors are incredibly immunosuppressive. Something like 40 to 50% of the tumor is macrophages, and there are no T cells. So I think the exciting opportunities with focused ultrasound are to modulate the immune system to deliver more T cells and to use other therapeutic approaches to eliminate all of that immunosuppressive environment. And I think that's going to be what's required to make a difference with immunotherapy in primary brain tumors.
And Patrick, you'll have the last word.
Quite the honor. Well, actually, I was impressed by the question from the audience about genetic engineering and using focused ultrasound for delivery. I noticed that there was a great presentation at this meeting from Beata Chertok and Dr. Rich Price. They had a mRNA delivery vehicle with the engineered nanoparticle, and then used the focused ultrasound to open those particles and to deliver the mRNA. And it was an mRNA for an anti-CTLA-4. So that technology to be able to really deliver our immunotherapy really precisely to where we want it, and then not have so many of the off-target effects, I think is really a potential exciting direction to go. just like tunable or switchable CAR T cells only being turned on right at the site of disease. I think that sort of precision is one of the advantages of focused ultrasound, is to more precisely control and minimize our off-target effects. So I hope those will be some of the things we will be hearing about at next year's meeting, and I hope we will have some updates on some of these ongoing clinical trials, and hopefully we will have some better understanding of what are some of the, as we've heard, the important impacts on the macrophages and on the cells in the microenvironment, because obviously that's a huge impact on where these treatments are going. So that's my hope for the next few years.
Okay, well, thank you. Well, thanks all of our panelists for this. It was a great discussion, and I look forward to future ones. So thank you.
Well, don't go anywhere just yet. Right before, we are going to take a break, and we'll be back here at 10:20 for the machine learning panel. But first, Matt Eames is going to come up, and he has a special presentation that he wants to make.
Hello. It's still the morning. Good morning. So I have a big picture of, nope, there it is, of Crit Moonen behind my head. I've been invited to spend a few moments recognizing Crit and his achievements. Rest assured, if it sounds this way, this is not an in memoriam. Crit is with us today. He's sitting right over there. Actually, Crit, if you would care to come over here and join me on stage, that would be wonderful. I'm going to talk about you as you walk over here. So I oversee the foundation's research funding, and so I thought it would be appropriate to start there because the foundation has a long history with Crit. We've actually, his first foundation-funded project was our 14th since opening our doors in 2016. And I guess I should say that to date, hi, welcome to the stage. To date, we've funded 239 research projects, and Crit is actually among one of the most recent as well that we've funded. He's been funded 4 times by the foundation, making him one of our 10 most frequently funded researchers. You didn't know that fact, did you? And his research projects with us have spanned from improving breast cancer ablation in collaboration with Mario Rees. He's touched on drug delivery with extracellular vesicles with us. And most recently, we're supporting his clinical endeavor into using histotripsy to modulate immune checkpoint inhibition to treat cancer, which of course is why I knew he'd be in this session to embarrass him. So just to say a bit more about Crit, he's had a career in focused ultrasound for over 2 decades. With his first focused ultrasound publications coming in 98, 99 in the areas of MR thermometry and FUS-induced transgene expression. In his long career, he's had over 250 publications. This spans both focused ultrasound and nuclear magnetic resonance. And if my math is correct, that makes for about more than 6 publications per year, which is pretty brisk pace in my opinion. And yours too, I'm sure. And let's see, in 2019, Crit was awarded the William and, I have to read this because it's a long title, the William and Frances Fry Honorary Fellowship for Contributions to the Field of Therapeutic Ultrasound, which is a Lifetime Achievement Award given by the International Society for Therapeutic Ultrasound. I guess I have a quote from Neil on the board behind me, that no one in the FUS community deserves more respect and admiration than Crit. He's the ultimate mentor and a role model that we should all try to emulate. Okay, why am I saying these things? So this summer, Crit reached out to the foundation and his Utrecht Center of Excellence colleagues to confess to us he has reached the mandatory retirement age in the Netherlands, but in the same sentence promised to continue his support of focused ultrasound in some way. He's clearly joined us for the symposium this week, which we appreciate, and we hope and expect to see you at future meetings. And so, if you haven't met Crit yet, I invite you to use the break to come introduce yourself. If you haven't yet caught up with him at this meeting, now's a great opportunity. And at this point, I'd like you all to join me in a round of applause for Crit's career and contribution in the field of focused ultrasound.
Oh, that's great, Matt. I'm very grateful to the society, to the foundation, I think, for the support over the years, and I hope to continue a little bit in a bit of a minor role, but still in a more modest role, but still to continue work and to support focused ultrasound. Thank you so much. Oh, that's really good.
My pleasure. Thank you. He didn't know I was going to do this. Anyway, we have a 30-minute break now, so we'll see you back here around 10:30. Thank you.